nucleic acid, you know that there are two types. Why they are known as nucleic acid? Because they are present in the nucleus, and they are the they contain, and in that you have phosphoric acid. That's why they are known as nucleic acid. DNA, deoxyribonucleic acid. DNA, then RNA, ribonucleic acid. In their character is acidic in nature because of that phosphate groups. Okay. That's why they are known as nucleic acid. We have seen about the DNA, and in the last session we have seen that Avery, McLeod, McCary, they demonstrated that DNA is a DNA cryptogenetic information. That experiment was proved by this Avery, McLeod, and McCary. DNA was discovered by Franklin Michel. Okay, Franklin Michel. We have seen what is in the nucleotides. Tides means three component: nitrogen, nitrogen base, pentose sugar, and phosphate group. In the nucleoside, साइड मोस्टली कितना होता है? दो साइड होता है, लेफ्ट साइड राइट साइड. तो साइड मतलब क्या? दो. दो मतलब क्या? एक नाइट्रोजनस बेस रहेगा, एक पेंटोस शुगर रहेगा. सो टी फॉर थ्री. थ्री मीन्स थ्री कंपोनेंट: नाइट्रोजन बेस, पेंटोस शुगर, फॉस्फेट. Sides mostly they are of two type, left side, right side. So nitrogen base plus pentose sugar. We have seen that. Okay, what are the examples of purines? Because they used to ask you, one of the following is not of purine found in the nucleic acid. So purine is nothing but the adenine, guanine. Pyrimidines are nothing but the three side. Pyrimidines are nothing but the three: cytosine, thymine, and uracil. This uracil is found in the RNA. Uracil is a cytosine. Uracil is a pyrimidine found in the RNA only. So in the DNA you have thymine. So in the RNA this thymine is replaced by uracil. Now most of the time is a single stranded. DNA is a double stranded. But in some viruses you will find RNA as the RNA as a double stranded. So mostly DNA double stranded होता है और RNA single stranded होता है. But exceptions होते हैं. There are certain DNA which are single stranded. There are certain RNA which are double stranded. But RNA normally single stranded होता है और DNA double stranded. So most commonly found in sugar deoxyribose D-ribose in the RNA, deoxyribose in the DNA. That's why deoxyribonucleic acid and ribonucleic acid. We have already seen this part. Now these nitrogenous bases, they are held together by phosphodiester bonds in the DNA RNA. Okay. Now sugars, they will bind to a nitrogenous base by beta N glycosidic bond. We have seen this part. So I'm just repeating it. I'm waiting for few more students. That's why. ये पॉइंट्स के ऊपर क्वेश्चन आ चुके हैं. न्यूक्लियोसाइड, न्यूक्लियोटाइड्स, ठीक है. प्यूरिन्स कौन से होते हैं, पिरिमिडिन्स कौन से होते हैं न्यूक्लिक एसिड में, शुगर कौन सा किसमें है, बॉन्ड कौन सा है, इसके ऊपर क्वेश्चन आ चुके हैं ऑलरेडी. वी हैव सीन व्हाट डू यू मीन बाय, है ना. नाउ डीएनए, आरएनए, दे आर ऑर्गेनाइज्ड इन द सेल. डीएनए इज द जेनेटिक मटेरियल व्हिच कैरीज द genetic information. Okay, now this DNA is wrapped around the nucleoproteins. In the nucleus you have nucleoprotein. Nucleus of the cell will have nucleoprotein, and in that commonly found protein is nothing but the histone. Now this DNA is wrapped around the histone and it is stored in the nucleus. Okay, so if you digest the nucleoprotein, you are going by nucleus digest. अगर nucleoprotein को digest करेंगे nucleus से, enzyme, you will get a nucleotides which has a three components. If you digest the nucleotides with the enzymes or chemicals, you will get a nucleosides. Nucleosides are having two components. Nucleosides में दो component होता है. Again, if you hydrolyze the nucleosides also, nucleoprotein को hydrolyze करेंगे nucleus से, you will get a nucleotides. If you hydrolyze nucleoproteins, you will get a nucleotides. Three components. If you hydrolyze the nucleotides, you will get a nucleosides which is having two components. If you hydrolyze nucleosides, you are going to get inorganic acid, that is phosphoric acid, then bases, purines and pyrimidines, and then you are going to get sugars. That is the evidence that nucleic acid has a purine, pyrimidine bases, ribose sugar, and phosphoric acid. We have seen the examples of purines and pyrimidines. In the purine, you will have structure like this. This is a structure of purine. So in that purine you have adenine and guanine. So these are the examples of purines. pyrimidine. Remember, purines are always named anti-clockwise. Anti-clockwise is बने nomenclature होता है. In pyrimids they are always numbered clockwise direction. Then we have seen what is meant by central dogma of life. Central dogma of life. So in the pyrimids we have cytosine, thymine, uracil. These three are the pyrimids. Central dogma of life means what? Formation of duplicate copy of DNA. Formation of duplicate copy of DNA is nothing but the replication. Formation of duplicate copy of DNA or copying of its own structure is known as a replication. For the cell division, for the multiplication, you need a DNA. DNA is a genetic material, you know that. So first DNA is replicated. So copies of DNA are made, identical copies of DNA are made, and then from DNA, RNA is synthesized. From DNA, RNA is synthesized. That process is known as a transcription. And from RNA, you are going to get lastly protein. So proteins are synthesized from RNA. That process is known as translation. We have seen the structure of DNA. DNA is double helix. Sugars found in the DNA also we have seen this structure. Okay, what is meant by tautomerism? We have seen. So purines and pyrimidines, guanine and pyrimidines. Adenine guanine जो होता है purines में, adenine will not show you tautomerism, but guanine and other thymine, cytosine and uracil. So all these are having ketone group. So this ketone group at a physiological pH, they are mostly in a keto form. That is the lactam form. This is known as the lactine form or enol form, keto-enol form, alcohol form and aldehyde form. So known as a keto-enol form or okay, keto-enol form of these nucleotides. They show isomerism, they show stereoisomerism, they show tautomerism. This is known as a tautomerism. Okay, we have seen the central dogma of life. Again, copying of DNA is known as a replication by the enzymes. DNA is converted into RNA, or synthesis of RNA from DNA is known as a transcription. Again, with the help of enzymes. Then RNA is expressed as a proteins. RNA is expressed as a protein by using the process known as a translation. Again, with the help of enzymes. Proteins may be either structural protein or functional protein. We have already seen. We have seen this nucleotide basis. Okay, we have seen this structure of DNA. So remember Chargaff's rule, Irving Chargaff in 1940. He proposed that the content of DNA. So whatever adenine, okay, are there, they are having exactly equal to the thymine. So adenine thymine content of DNA is equal to the guanine cytosine. Okay, as like that. जितना DNA में adenine होगा, उतना thymine होगा. जितना डीएनए में कॉनिन होगा उतना ही क्या होगा साइटोसिन. It is known as a molar equivalence of purines and pyrimidines. So number of purines which are equal to the number of pyrimidines in the DNA structure known as a molar equivalence. Chargaff's rule of molar equivalence between purines and pyrimidines. Now it is in the DNA. DNA में ये होता है because DNA is double stranded. This rule is not followed by RNA. Chargaff's rule is not followed by RNA. It is only followed by double-stranded DNA. Double-stranded DNA. Double-stranded DNA. Also, this rule is not followed by single-stranded DNA. जो double-stranded DNA होता है वही rule follow करेगा ये. मतलब rule क्या है? Number of purines is equal to number of pyrimidines. So number of adenine and is equal to number of thymine. Number of guanine is equal to number of cytosine. That rule is followed by double-stranded DNA structure, not followed by single-stranded DNA structure, and also not followed by your RNA structure. Double-stranded RNA also sometimes follow this rule. We have seen this. After that, we have seen the structure of DNA also. Types of types of DNA also. So bDNA is the most common. bDNA is the most stable structure, which is known as a Watson and Crick model. Okay. After that, okay, A to E. You know, there are different types of DNA. You know, ranging from DNA A, B, C, D, E. You know, so almost there are 6 types of DNA. Okay, from A to E. But most common form of DNA is nothing but the bDNA. इसके ऊपर question आ चुका है already. So bDNA is the most common form, which is known as a Watson and Crick model. Now aDNA is nothing but the bDNA is a left-handed DNA. Remember. Then aDNA is a right-handed DNA. zDNA is also left-handed DNA. because in the zDNA the two polynucleotide chains, they will move in a zigzag manner. That's why they are known as zDNA. Remember, bDNA is the most prominent under physiological condition. Denaturation of DNA means loss of native structure of or loss of helical structure of DNA. Melting temperature क्या होता है, इसके ऊपर क्वेश्चन आ चुका है. Melting temperature means it is a temperature, okay, temperature at half of the helical structure will be lost. Question was there. Okay? We have seen. Okay? Now structure of RNA. DNA का structure आपने देखा है. Am I audible to you? Now structure of RNA. Again, as the DNA is nothing but the DNA is nothing but the polymer of deoxynucleotide. Okay, deoxyribonucleotide. Okay. In case of DNA, deoxyribonucleotide, because DNA मतलब deoxy sugar होगा उसमें. So DNA is a polymer of deoxyribonucleotides. But in case of RNA, RNA is a polymer of ribonucleotides. Remember, RNA is a polymer of ribonucleotide. Now it is also held together by 3'5' phosphodiester bond. It has a difference. DNA में और RNA में क्या difference है? So in the DNA you have deoxyribose sugar. So here you will have ribose sugar. So first difference in the sugar. Second difference. So in the base, in the pyrimidine base, instead of thymine, so DNA has a pyrimidine. Okay, DNA में क्या है कि thymine होता है pyrimidine. In the DNA, pyrimidine is thymine, but in the RNA it is a uracil. Now usually RNA are single stranded, DNA double stranded होता है. RNA are a single stranded, and they will not follow or they will not obey Chargaff's rule. Again, RNA is susceptible for the alkaline hydrolysis, and they will also give you color reaction with the orsinol. Orsinolic color reaction देता है. It is orsinol is a chemical which react with the ribose sugar, okay, in the RNA, and it will give you some color. So these are the features of your RNA. RNA क्या होता है? RNA is nothing but single stranded. It is a chain or it is a polymer of ribonucleotide. These held together by 5'3' phosphodiester bond. Then RNA has a ribose sugar, DNA has a deoxyribose sugar. DNA is a double stranded, RNA is single stranded. In the DNA, Chargaff's rules are followed. In the RNA, Chargaff rule is not followed. Most of the time, RNA is a single stranded, but there are certain viruses which also have genetic material as RNA, which are double stranded. And that in that condition, it may follow Chargaff's rule. Types of RNA, there are different types of RNA, and these RNA will have different functions. So first type is nothing but the messenger RNA. So messenger RNA is about 5 messenger RNA is a 5 to 10% of total RNA. And what is the function of messenger RNA? As the name indicate, messenger. मैसेंजर का काम होता है कि आपको मैसेज देना है ना. आप फेसबुक के ऊपर मैसेंजर यूज करते हो, उसका काम क्या होता है कि किसी ने आपको मैसेज भेजा तो आपको मैसेज मिल जाएगा. तो ये मैसेंजर RNA का काम भी वही होता है. सो सेम वर्क इज देयर, सेम रोल इज देयर. सो व्हाट इज द रोल ऑफ मैसेंजर RNA? सो इट विल मैसेज. So it will message. What message? So message present on the DNA that will be transferred to the protein synthesis assembly. So codings are there on a mRNA. Different codings which will code for a particular amino acid, right? So that message is there on a DNA. Whatever the genetic information is there in the DNA, that is copied on a mRNA. And mRNA is nothing but the messenger RNA. So that message from DNA to the okay, that copy, it is a copy of DNA. Simply, mRNA is a copy of DNA, and message from DNA is copied in the mRNA, and mRNA will transfer this message to a protein synthesis assembly, and according to that only the proteins will be synthesized. So messenger RNA comprises about 5 to 10%. Then second type होता है आपका RNA का ट्रांसफर RNA. So as the name indicate, ट्रांसफर RNA, इसका काम होता है ट्रांसफर. इसका ट्रांसफर, so it will transfer amino acid. It will transfer amino acid. So it will take one amino acid along with this tRNA. Amino acid will bind to tRNA, and it will be transferred to a protein synthesis assembly according to the codes present on the mRNA. mRNA के ऊपर कोड्स होंगे, और जो कोड रीड होगा उसके अकॉर्डिंगली ये अमीनो एसिड लेके आएगा. कौन? ट्रांसफर RNA. Now next is the which is a 10 to 20%. Transfer RNA is a 10 to 20%. Third one is the ribosomal RNA. Ribosomal RNA will comprise around 50 to 80%. Now, ribosomal RNA, it is the structural framework where there is a protein synthesis. Protein synthesis कहाँ पे होता है? Ribosome में होता है. Ribosomal RNA. They are the component of ribosome. So okay, so they are the site for synthesis of proteins. Ribosomal RNA, they are the site for synthesis of proteins. Now messenger RNA will carry a code. Messenger RNA will carry a message according to the DNA. Messenger RNA will carry a code according to the DNA. Transfer RNA will pick up one amino acid or will transfer one amino acid and according to the code present on mRNA. So जो कोड होता है mRNA के ऊपर वो रीड किया जाता है और उसके अकॉर्डिंगली ट्रांसफर आने का काम होता है कि आप वो अमीनो एसिड लेके आओ. वो अमीनो एसिड लेके आएगा ट्रांसफर आने इसके बेसिस पे mRNA के बेसिस पे और वो बाद में असेंबलिंग पेप्टाइड बॉन्ड फॉर्मेशन कहाँ पे होगा. So there is a formation of peptide bond in this ribosomal RNA. So RNA, these are the types of RNA. We have seen already the structure of DNA. Again you will see the structure of DNA. DNA is a double stranded. DNA is a double stranded. Okay. So there are two polydeoxyribonucleotide chain A and chain B. Okay. Now these chains, they are twisted along with each other. They are twisted with each other, and that's why it look like a twisted ladder. Okay. Now base pairs. Now this backbone of this nucleotide is nothing but the phosphodiester bond. Phosphodiester bond. ये जो backbone है वो phosphodiester bond है, and in between this bond, so in between the pyrimidine thymine guanine, ये जो bonding है in between this the base pairs are there. So the bonding between base pair, that is nothing but the hydrogen bond. So there is a hydrogen bond between base pair. Now remember, for the purines will form a two hydrogen bond. Purines will form a two hydrogen bond, and pyrimidines will form a three hydrogen bond. Pyrimidines will form a three hydrogen bond. Purines will form a two hydrogen bond, and pyrimidines will form a three hydrogen bonds. तो आपको ये याद रखना है, right? In case of DNA. So thymine, cytosine, adenine, guanine. These are the commonly nitrogenous bases found in the DNA. Now RNA is a single stranded, as you can see over here. This is the structure of RNA. RNA is a single strand. Again the backbone is nothing but the backbone in the RNA structure is nothing but the phosphodiester bond. So what are the base pairs which are present in the nitrogenous bases present in the RNA? So here you will have guanine, adenine, cytosine and uracil. Instead of thymine, you have uracil in the RNA. Now we have seen these for metabolism of these nucleotides. Every nucleotide will have its own metabolism. इसका metabolism अलग-अलग होता है हर एक nucleotide का, है ना. Thymine का अलग है, cytosine का अलग है ना. So if you want to see the individual metabolism, you can read from books. Actually, individual metabolism of these nucleotides, individual metabolism of amino acids, they are not important. That's why I have skipped that part. Okay? So they are not important over here. Okay, but definitely as far as the amino acid concerned, their inborn errors we have taken because they are important, for example. But individual amino acid metabolism, that is very tedious. If you want to go in it, it will take at least 2-3 lectures. And again, individual metabolism of these nucleotides will also take at least 2 lectures. That's why I have skipped that part. Remember, in the RNA, uracil is there instead of thymine. RNA is a single stranded, right? Now, there are types of RNA. There are different types. Okay, there are different types. Now you can see over here. So different types of DNA also. Comparison of structural feature of different types of DNA. Structural feature and different conformation of DNA double helix. DNA का भी structure जो है different होता है. जैसे most common जो होता है BDNA, which is right handed, है ना. ADNA भी होता है, then ZDNA भी होता है. So here feature, so helix types. So BDNA is a BDN is the most common and is right-handed. Okay, it is a Watson and Crick model. यही को इसको ही बोलते हैं Watson and Crick model, right? Then ADNA, Watson and Crick model, the BDNA. Now, diameter of this Watson and Crick model of DNA is nothing but the 2.37 nanometer or 2.3 okay angstrom. Now, distance per each complete turn is distance of each groove. So it is a 3.4 nanometer or 34 angstrom. जो so ये जो है ग्रूव मेजर ग्रूव माइनर ग्रूव. So distance per each complete turn, ये जो टर्न होता है इसका डिस्टेंस कितना होता है 3.4 नैनोमीटर और 34 एंगस्ट्रॉम फॉर द BDN. नाउ rise per base pair. Rise per base pair means the distance between two base pair is nothing but the 0.34 nanometer, or you can say 3.4 angstrom or 3.4 angstrom. Number of base pairs in each turn. Number of base pairs in each turn for BDNA is around 10 base pair. तो BDNA जो होता है इसमें एक ग्रुप में 10 बेस पेयर होंगे. अब बेस पेयर टिल्ट मतलब उसका एंगल 19 होता है. हेलिक्स एक्सिस ऑफ रोटेशन मेजर ग्रुप, ये मेजर ग्रुप रहता है इसमें, राइट. नाउ अनदर टाइप इज ADNA व्हिच इज राइट हैंडेड DNA. इसमें क्या रहेगा, हेलिकल का डायमीटर जो होता है, डायमीटर ऑफ हेलिक्स इज अराउंड 2.55 नैनोमीटर और इवन से 25 angstrom. 2.55 nanometer or 25 angstrom. Then each turn or each groove will have a distance of 3.2 nanometer or 32 angstrom. Okay, distance between each base pair in the ADNA is around 0.29 angstrom or you can say 0.29 nanometer or 2.9 angstrom. Then number of base pairs per turn in each group. So in case of ADNA you have 11 base pairs ना. जो मेजर जो Watson and Crick मॉडल है उसमें कितना बेस पेयर होता है 10 बेस पेयर, बट ADNA में 11 बेस पेयर होता है. देन यू कैन सी बाकी ये दोनों छोड़ दो. इन केस ऑफ ZDNA the diameter of helical structure, so it is a 1.84 nanometer or you can see है ना, you can see 18 angstrom है ना, 18 angstrom or distance between each complete turn. So it is the distance of each groove is a 4.5 nanometer or 45 angstrom. देखो BDNA का जो मेजर ग्रुव है उसका जो डिस्टेंस है 34 angstrom और 3.4 नैनोमीटर. ADNA का 32 angstrom और 3.2 नैनोमीटर और जो ZDNA होता है उसका 45 angstrom और 4.5 नैनोमीटर. ठीक है, तो मतलब ZDNA का ग्रुप बहुत बड़ा है. Number of distance between each base pair. So in case of BDNA 0.34 nanometer मतलब 3.4 angstrom. 0.29 in case of ADNA मतलब है ना 2.9 angstrom and distance between each base pair in the ZDNA is 0.37 nanometer or 3.7 angstrom. Now in case of base pairs, number of base pair in the each group. In case of BDNA there are 10 base pairs in each group. In case of ADNA 11 base pairs and ZDNA में होता है 12 base pairs. So that is a comparison between three structure of DNA. These are the mostly common. But there are A, B, C, D, E. There are at least 5 types of DNA. We'll see the structure of DNA. Structure of RNA. So there are 3 types of RNA. RNA में 3 टाइप होता है. पहला होता है messenger RNA, mRNA. This one is a mRNA. This one is a ribosomal RNA, and third one is a tRNA, transfer RNA. Okay, so RNA they are single stranded. Again, they are the polymer of ribonucleotides. They are the polymer of ribonucleotides held together by phosphodiesterase bond. mRNA, that is known as a messenger RNA, and the role is nothing but to carry the message from DNA to the protein synthesis assembly. So whatever message, whatever genetic information is present on the DNA, that is copied in the mRNA in the form of codes. Okay, now that mRNA will transfer that codes to the protein synthesis assembly and according to the code which are present on the mRNA. So tRNA is known as a transfer RNA, and the role of tRNA is nothing but to take amino acid along with it. So it transfers the amino acid for the protein synthesis. So this code on mRNA will be read, and according to that, this tRNA will carry one amino acid. Right? Now the protein synthesis is done in the ribosomes. It is the structural framework for protein synthesis. Now whatever codes are present on the mRNA, according to that, amino acids are carried, and there is a formation of peptide bond in the ribosome known as a protein synthesis. So mRNA structure is a linear. It is a linear structure. Okay, the synthesis of mRNA from the DNA known as a transcription. Okay, replication means formation of copies of DNA known as a replication, and from the DNA RNA is synthesized known as a transcription, and from that RNA proteins are synthesized known as a translation. Right, ribosomal RNA, mRNA and tRNA. Now tRNA has a, it will look like a clover leaf. जो tRNA होता है ना, वो clover leaf के जैसे होता है. इसके ऊपर क्वेश्चन आ चुका है. Which RNA will have structure looking like a clover leaf? tRNA will look like a clover leaf. You can see this is a tRNA structure. Again, in the tRNA you have different type of arms. Okay, different clover leaf is called the clover leaf structure. Okay, you have acceptor arm, that is known as acceptor arm, which accepts the amino acid or carries the amino acid. So amino acid will bind to a 3' chain. Okay. Then you have anticodon arm, which is these anticodon means these are the complementary codon to this mRNA. mRNA के ऊपर जो 3 codon रहेंगे, UAG, UGA, ऐसा, उसके complementary codon यहाँ पे tRNA मिलेंगे, so that to read a particular amino acid, right? Then एक another arm होता है THU arm, और एक short arm होता है यहाँ पे, and another arm is there, pseudouridine arm or pseudouridine arm, psi arm. You see in the detail, just structure lower. So remember, tRNA will look like a clover leaf model. Clover leaf. Types of messenger RNA now. Types of messenger RNA. Type of RNA, their function. Okay. Type of RNA and their function. So पहला होता है आपका messenger RNA. So what is the function of mRNA? It transfers the genetic information from genes to the ribosome for the protein synthesis. I have told you. Okay. Now eukaryotic, there are two cells, prokaryotic cell and eukaryotic cell. दो टाइप के सेल होता है, prokaryotic cell and eukaryotic cell. So prokaryotic cell means cells of microorganism. Cell of microorganism known as a prokaryotic cell, and eukaryotic means cell of humans, okay, or higher animals. So prokaryotic and eukaryotic. So there is a little bit difference in the prokaryotic cell, prokaryotic cellular process and eukaryotic cellular process. That's why antibiotics they can kill selectively and microbials and will not harm to the host cell. There is a difference between the replication process of prokaryotic cell, prokaryotic DNA, transcription of prokaryotic DNA, and protein synthesis in the prokaryotic cell. So there is a different enzymes, different steps, and that's why in the prokaryotic replication you have different enzymes. That's why antibacterial drug will particularly kill the bacteria, not our human cell, because there is a difference. There is a definite difference in the replication process, replication steps, replication enzyme of the bacterial DNA. There is difference between bacterial transcription, the enzymes required for the transcription in the bacteria, and in the eukaryotic cell. There is a difference between the steps in the protein synthesis, the enzymes required for the protein synthesis in the bacteria and in the eukaryotic cell. So that's why antibiotic they can target selectively on the bacterial cell. Now eukaryotic mRNA is capped at a 5' terminal and by 7-methylguanosine triphosphate. जो आपका ह्यूमन का mRNA होता है, उसका जो 5' एंड होता है, on that you will find 7-methylguanosine triphosphate, which is a, you can say it is a cap. It is capped with the 7-methylguanosine triphosphate. इसके ऊपर भी क्वेश्चन आता है. Eukaryotic mRNA is capped at a pyridase terminal by 7-methyl traps, which prevent mRNA from hydrolysis. Which prevent the mRNA from hydrolysis. Okay. Types of RNA. टाइप्स ऑफ आरएनए इंपॉर्टेंट है, क्वेश्चन ऑलरेडी आ चुका है इसके ऊपर। सेकंड टाइप होता है हेट्रोजिनस न्यूक्लियर आरएनए। हेट्रोजिनस न्यूक्लियर आरएनए एचएनआरएनए, इसके ऊपर क्वेश्चन आ चुका है। एचएनआरएनए हेट्रोजिनस न्यूक्लियर आरएनए, व्हाट इज द रोल ऑफ दिस हेट्रोजिनस न्यूक्लियर आरएनए? मैसेंजर आरएनए का काम होता है कि मैसेज देना। किससे मैसेज देना है भाई? जो भी डीएनए के ही अकॉर्डिंगली, डीएनए से ही एमआरएनए बनता है ट्रांसक्रिप्शन प्रोसेस से। तो जो जेनेटिक इंफॉर्मेशन डीएनए के ऊपर है वही कॉपी होके एमआरएनए पे आ गया, ठीक है। और एमआरएनए के अकॉर्डिंगली वो प्रोटीन बनेगा, राइट। तो स्पेसिफिक प्रोटीन विल सर्व अ स्पेसिफिक फंक्शन, है ना। अगर कोई फॉल्टी प्रोटीन बनता है तो आपको फिर इनबॉर्न एरर हो जाता है। इफ देयर इज अ फॉल्टी प्रोटीन देन यू विल सफर फ्रॉम जेनेटिक डिसीज। हेट्रोजेनस न्यूक्लियर आरएनए एचएनआरएनए serve as a precursor for messenger RNA and other RNAs. Yeah, what is the role of this heterogeneous nuclear RNA HNRNA? It is a precursor for mRNA and other RNA. Precursor होता है. Next one is a transfer RNA or not also known as a tRNA, also known as a soluble RNA. Now tRNA will look like a clover leaf. tRNA will look like a clover leaf. इसके ऊपर क्वेश्चन आ चुका है. What is the role of tRNA? As the name indicates, T is transfer. tRNA is involved in the transfer of amino acid to mRNA for protein synthesis. It consists of different arms. There are 4 arms. इसमें 4 आर्म होते हैं. पहला आर्म होता है आपका acceptor arm. So acceptor arm, it is a acceptance site for the amino acid. Now this arm is capped with the CCA 5' end. तो जो 5' end होता है ये आर्म में, उसके ऊपर ये 3 न्यूक्लियोटाइड बेस होते हैं ना, साइटोसिन एंड एडेनिन, CCA. So acceptor arm is capped with the CCA at 5' to 3'. The amino acid is attached to a acceptor arm. The second होता है anticodon arm. So containing 3 specific anticodon which are responsible for the recognition of triplet codon on the mRNA. जो है ना, एंटीकोडन आर्म जो होता है उसके ऊपर अपोजिट कोडन होते हैं जो 3, जो mRNA के ऊपर जो कोड रहेगा उसके अपोजिट कोड रहेंगे। तो जब वो मैच होंगे, वो जब रीड करेंगे, रिकॉग्नाइज करेंगे, तब वो अमीनो एसिड लेके आएंगे। देन okay, so acceptor arm to attach the amino acid, anticodon arm to recognize the triplet codon on the mRNA, and third arm होता है the D arm. So D arm is the name given because of the presence of dihydrouridine. Then fourth arm होता है T-Cy arm, which consists of a sequence known as a pseudouridine. And the variable arm होता है because of the variability of tRNA. They are classified as a tRNA 1, 2, etc. 1, 2, 3, 4 tRNA. Then next होता है ribosomal RNA. Ribosomal RNA is known as a rRNA. So they provides a structural framework for ribosomes. Now GPAT 2019 में ये क्वेश्चन आ चुका है. Small nuclear RNA. What is the role of small nuclear RNA? Small nuclear RNA, snRNA. Small nuclear RNA, snRNA. Small nuclear RNA, snRNA. So it is involved in the processing of mRNA. ये mRNA का प्रोसेसिंग करता है. ये mRNA का प्रोसेसिंग कौन करता है? स्मॉल न्यूक्लियर RNA, snRNA. इसके ऊपर क्वेश्चन GPAT 2019 में आ चुका है, snRNA. So during the synthesis of mRNA, जब DNA से, है ना, DNA डबल स्ट्रैंड होता है, DNA से क्या बनता है आपका RNA क्या आता है? सिंगल स्ट्रैंड. So during the transcription process, so whether mRNA is synthesized in a proper way or whether it is correct or not, so that is done by the small nuclear RNA, snRNA. So snRNA has a role in the processing of mRNA. mRNA को प्रोसेस कौन करेगा? snRNA. इसका काम पूछा था GPAT 2019 में. Am I audible to you? Okay. Then small nucleolar RNA, स्मॉल न्यूक्लियर आरएनए एसएन आरएनए, स्मॉल न्यूक्लियोलार आरएनए, तो यहाँ पे एसएन ओ डाल दो, न्यूक्लियोलार, न्यूक्लियोला, न्यूक्लियर, सो न्यूक्लियर मतलब एसएन आरएनए, न्यूक्लियोलार मतलब एसएन ओ आरएनए. So it has a play, it plays very important role in the again processing of mRNA molecule, plays a key role in the processing of mRNA molecule. Again same thing like that, nuclear RNA. जैसे न्यूक्लियर आरएनए काम करता है, प्रोसेसिंग का वही काम कौन करता है, जैसे नो आरएनए. देन अगेन नेक्स्ट टाइप होता है स्मॉल साइटोप्लाज्मिक आरएनए, स्मॉल साइटोप्लाज्मिक आरएनए एसओ आरएनए. इट इज इन्वॉल्व इन द सिलेक्शन ऑफ प्रोटीन फॉर एक्सपोर्ट. जब प्रोटीन सिंथेसिस होगा उसके बाद में जो प्रोटीन का पोस्ट ट्रांसक्रिप्शनल मॉडिफिकेशन होगा, तो इनिशियली जो प्रोटीन सिंथेसिस होता है दैट इज अ रॉ प्रोटीन, दैट इज अ नॉन फंक्शनल प्रोटीन, दैट इज अ नॉन फंक्शनल protein, but that protein is made functional. Okay, that protein is made functional by post transcriptional modification, and after the modification that protein is exported, that protein is exported, so that protein is exported after functional protein is synthesized, that protein is exported out of the cell. Now that is done by small cytoplasmic RNA, SORNA. Next is nothing but the transfer messenger RNA, tmRNA, transfer messenger RNA, tmRNA, transfer messenger RNA. So it is mostly present in the bacteria, and its function is nothing but the addition of short peptide tags to the protein to facilitate the degradation of incorrectly synthesized protein. तो ड्यूरिंग द प्रोटीन सिंथेसिस क्या होता है कि कभी कभी इनकरेक्ट प्रोटीन सिंथेसिस होता है ड्यू टू म्यूटेशंस, ड्यू टू समथिंग, सम एरर्स इन द सिंथेसिस. तो वो जो प्रोटीन गलत सिंथेसिस हो गया तो उसको रखेंगे तो फिर डिस हो जाएगा, वो एंटीजन बन जाएगा, एटोमिक डिस हो जाएगा. सो व्हाट, सो दैट प्रोटीन शुड बी डिग्रेडेड. सो फॉर दैट दिस आरएनए इज देयर, ट्रांसफर मैसेंजर आरएनए, टीएमआरएनए, which will cause degradation, which can degrade the incorrectly synthesized protein in the bacteria. So they used to ask you same thing, what is the role of different RNA, हर एक RNA का रोल क्या है, इम्पोर्टेंट है ये, इसके ऊपर क्वेश्चन आता है. तो mRNA देखो, heterogeneous nuclear RNA देखो, RNA, ट्रांसफर RNA, then नेक्स्ट आता है आपका राइबोसोमल RNA, then small nuclear RNA, snRNA, small nucleolar RNA, snRNA. तो ये दोनों भी न्यूक्लियर RNA जो होता है, न्यूक्लियर RNA और न्यूक्लियोलार RNA, so it is in the processing of mRNA. बाद में small cytoplasmic RNA, selection of protein for export, and बाद में transfer messenger RNA, so that will degrade the incorrectly synthesized protein in the bacteria. Now you can see over here again same thing, रिवाइज करो, मैसेंजर आरएनए ट्रीटेड एज एमआरएनए, मैसेंजर आरएनए को ऐसे लिखते हैं एमआरएनए, राइट. सो काम क्या है इसका, व्हाट इज फंक्शन, ट्रांसफर द जेनेटिक इंफॉर्मेशन फ्रॉम जीन्स टू द राइबोसोम टू सिंथेसाइज द प्रोटीन. नाउ हेटेरोजेनस न्यूक्लियर आरएनए, एचएनआरएनए, सो इट इज अ प्रीकर्सर फॉर सिंथेसिस ऑफ एमआरएनए एंड अदर आरएनए. ट्रांसफर आरएनए, टीआरएनए बोलते हैं उसको, ट्रांसफर द अमीनो एसिड फ्रॉम एमआरएनए टू द प्रोटीन सिंथेसिस. नेक्स्ट होता है राइबोसोमल आरएनए, उसको अपन आरआरएनए बोलते हैं. सो इट प्रोवाइड्स द स्ट्रक्चरल फ्रेमवर्क फॉर द राइबोसोम. नेक्स्ट होता है स्मॉल न्यूक्लियर आरएनए, एसएनआरएनए, इट इज नोन इन द एमआरएनए प्रोसेसिंग. स्मॉल न्यूक्लियोलार आरएनए, एसएनओआरएनए, इन्वॉल्व इन द प्रोसेसिंग ऑफ एमआरएनए. क्वेश्चन इसके ऊपर आ चुकी है. स्मॉल साइटोप्लाज्मिक आरएनए, SCRNA, involved in the selection of protein for export. Transfer messenger RNA, tmRNA, degradation of incorrectly synthesized protein. याद रखना है क्वेश्चन आता है इसका. सो इंडिविजुअल जो न्यूक्लियोटाइड्स होता है, इंडिविजुअल न्यूक्लियोसाइड्स हैं, मेटाबॉलिज्म ऑफ इंडिविजुअल न्यूक्लियोटाइड्स इज नॉट possible to take because that will take a lot of lectures, but okay, so you should know, if you want to read, you can read. So there is some nucleotide known as xanthine and hypoxanthine, nitrogenous bases xanthine and hypoxanthine. So metabolism of these, lastly they will produce a uric acid. Lastly they will produce a uric acid. And if there is increased uric acid in the body, okay, normally uric acid is kept okay, kept in a solution. But if at extremities है ना, जैसे आपका पैर होता है, जैसे हाथ होते हैं, so in the extremities you have less blood flow, and again and you have less body temperature over there है ना. आपके पैर के जो देखो आप टो, पैर का टो जो होता है वहां पे बॉडी टेम्परेचर कम होता है, राइट. सो डिक्रीज इफ देयर इज अ लेस बॉडी टेम्परेचर, दिस एक्सेसिव यूरिक एसिड, जो यूरिक एसिड बढ़ा है, तो यूरिक एसिड क्या होता है, नॉर्मली इट इज इन अ सॉल्यूशन इन द ब्लड, बट व्हेन at the extremities, for example toes, knees, etc, etc, so what happens, due to decreased temperature, now these uric acid they get crystallized, uric acid get crystallized and they get deposited on the toes and joints, right, and they causes the inflammation, pain and all that. That is known as a arthritis, that is known as a gout. Okay, now same uric acid get deposited in the kidney, okay, and causes the kidney stone also. Okay, so that's why this is denoted word. So adenine monophosphate जो होता है न्यूक्लियोटाइड, उससे बनता है इनोसिन मोनोफॉस्फेट, और उससे बनता है इनोसिन, इनोसिन से बनता है हाइपोजैंथीन, हाइपोजैंथीन से बनता है जैंथीन, और जैंथीन से लास्ट बनता है यूरिक एसिड. Increased uric acid level in the body leads to gout and arthritis. Also it get deposited in the kidney and causes the kidney stone. यूरिक एसिड, सोडियम सॉल्ट ऑफ यूरिक एसिड, सोडियम यूरिएट, पोटेशियम यूरिएट, वो सब डिपोजिट होगा किडनी में और करेगा कैलकुलाई और स्टोन्स. सेम यूरिक एसिड crystal deposited in the joints causes the gout. Okay, now allopurinol is one drug. Allopurinol एक drug होता है ना, allopurinol. So that drug inhibit the xanthine oxidase enzyme. ये जो enzyme है xanthine oxidase, that is inhibited by this allopurinol, and allopurinol prevent the synthesis of uric acid, and that's why allopurinol is used in the treatment of gout. Biochemistry is very important. Biochemistry is the basis for your pharmacology. HAP is the basis for your pharmacology. Are you getting? Remember this. Okay. And remember we have seen in the enzymes also. Allopurinol is a allosteric inhibitor of xanthine oxidase. Allopurinol is a allosteric inhibitor of xanthine oxidase. So we have seen in the enzyme what is meant by allosteric inhibition. So allosteric inhibitors, they will bind to the another site known as allosteric site of enzyme. Now major classes of proteins, their function involved in the DNA replication, DNA transcription, etc, etc. So DNA replication में क्या क्या लगता है आपको? Replication of DNA means formation of identical copies of DNA, and for that you need a certain enzymes, you need a certain proteins, you need a certain protein. So before that, what is DNA? DNA is nothing but the polymer of your deoxyribonucleotide. DNA is nothing but the polymer of your deoxyribonucleotide. So in the DNA, these nucleotides, they are arranged in this fashion. Nucleotides are arranged in this fashion. जैसे यहाँ पे है ना, ये क्या है, ये पेंटो शुगर है, दिस इज पेंटो शुगर, दिस इज पेंटो शुगर. सो थायमिन इज न्यूक्लियोटाइड बेस. थायमिन, यहाँ पे थायमिन रहेगा, उसको ये थायमिन न्यूक्लियोटाइड बेस प्लस पेंटो शुगर नोन एज अ न्यूक्लियोसाइड. ओके, now pentose sugar, यहाँ पे कार्बन नंबर 1 है 2 4 and this is a 5, carbon number 5. Now add the carbon number 5 of pentose sugar, there is a attachment of phosphate group, and this end is known as a phosphate 5' phosphate end. Now third carbon of your pentose sugar, again there is a phosphate bond formation, phosphate diphosphate bond, another nucleotide cytosine. Okay, so in this similar way again diphosphate bond in this. Okay. Now this, the OH group, third carbon atom of sugar, okay, known as 3' end, 3' end, and this is known as a 5' end. This is known as a 5' end. Why 3' 5' end? Because 5' end means the फॉस्फेट, ओके, फॉस्फेट बॉन्ड का ग्रुप जो होता है फाइडेस्ट वही चीज नोन एज फॉस्फेट फॉस्फेट एंड 3 डैश क्या होता है, तो थर्ड कार्बन ऑफ पेंटोस शुगर OH group is there known as 3 dash, right? DNA is nothing but the polymer of deoxyribonucleic acid, deoxyribonucleotides, deoxyribonucleotide ये इनका पॉलीमर है and this is known as a phosphodiester bond, phosphodiester bond जो है इसको बोलेंगे अपन phosphodiester bond. Now what are the major classes of proteins required for the replication and they are involved in the replication? DNA polymerase, DNA polymerase पहला प्रश्न होता है DNA polymerase. So there are different isoenzymes of DNA polymerase. इसमें types of isomer enzyme होता है. So DNA polymerase type 1, DNA polymerase type 2, DNA polymerase 3 ऐसा होता है. DNA polymerase 3. So there are 3 types: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Okay. What is the function of DNA polymerase in the replication? So as the name indicates, polymerase. Poly मतलब क्या, बहुत सारा, मरेज मतलब क्या, मर्ज करना. तो बहुत सारा मर्ज करता है, बहुत सारा क्या मर्ज करता है, न्यूक्लियोटाइड मर्ज करता है भाई. तो न्यूक्लियोटाइड they are joined together by DNA polymerase, right? So according to the parent strand, so whatever template strand is there on a DNA, so in complementary, okay, in complementary to that strand, so DNA polymerase will add a nucleotide and will form a new strand, okay? Because replication of DNA मतलब क्या है कि DNA का आइडेंटिकल कॉपीज बनना है और उसके लिए आपको एंजाइम चाहिए, उसके लिए आपको न्यूक्लियोटाइड चाहिए, उसके लिए आपको प्रोमोटर चाहिए, इनहिबिटर चाहिए, लाइक दैट. So DNA polymerase का टाइप होता है टाइप 1, टाइप 2, टाइप 3. So what is the function? Polymerization of deoxyribonucleotide. Polymerization of deoxyribonucleotide. Now इसके जो मैंने बोला टाइप 1, टाइप 2, टाइप DNA polymerase 1, 2, 3. So DNA polymerase 3 का काम होता है to add the nucleotides. DNA polymerase 1 का काम होता है, DNA polymerase 1 and 2 का काम होता है repair और proofreading. Repair मतलब क्या, if any false base pair is added, so that is removed. So repair enzyme कौन है, DNA polymerase 1 and 2. DNA polymerase 3 क्या है, it will polymerize the nucleotide and also has a proofreading activity. बाद में देखें. Now another enzyme is DNA helicase. DNA helicase, what is the role of DNA helicase? So unwinding of DNA. DNA का स्ट्रक्चर कैसा है, डबल हेलिकल है. तो डबल हेलिकल स्ट्रक्चर का तो एज इट इज कॉपी नहीं बना सकते, सो यू हैव टू अनवाइंड दैट. सो डबल हेलिकल स्ट्रक्चर जो है इट इज broken down, that is broken down at some place. So DNA helicase का काम क्या होता है, what is the role of DNA helicase? So it will cause unwinding of DNA. So जैसे zip होता है आपका, पैंट का zip होता है, तो zip ओपन करने के लिए क्या है, zipper होता है ना, क्लोज करने के लिए zipper. So DNA helicase will act like a same way. So it is a zip opener. You can say it is a zip opener. So it will open a two-stranded DNA. It will open the double-stranded DNA for the replication. Now another enzyme is there known as a DNA primase. तो उसका काम क्या है, DNA primase. नाम में रखा है, बोलते नाम में क्या रखा है, primase मतलब ये क्या करेगा, प्राइमर बनाएगा. So it initiate the synthesis of RNA primer. Because for the DNA replication कुछ तो भी स्टार्टिंग पॉइंट चाहिए ना, स्टार्टिंग के लिए ना किक चाहिए. तो बोलते हो पढ़ना है लेकिन पढ़ने के लिए कुछ किक मिलना चाहिए, वैसे ही डीएनए का रेप्लिकेशन करने के लिए कुछ इनिशियल चाहिए, कुछ इनिशियल बेस पेयर, से फॉर एग्जांपल 15 to 20 बेस पेयर. So they are required. So it is nothing but the RNA primer. So short fragment of nucleotides, they are synthesized by primase enzyme known as the RNA primer. Now this RNA primer will attach to a one strand and will add the nucleotides in complementary to the parent strand. Okay. Then next होता है topoisomerase. Next enzyme है topoisomerase. टोपो मतलब क्या, टोपी मतलब क्या, टोपी किसमें होता है, टोपी आप काय के लिए यूज करते हो, जब है ना, टोपी क्यों यूज करते हो, जैसे अपना पूरा सर है, पूरा टकला है, वो पूरा हाइड करने के लिए टोपी यूज करते हो, तो सील करने के लिए यूज करते हो. व्हाट इज द रोल ऑफ टोपोइसोमरेज? nick sealing enzyme. It relieves the torsional strain resulting from the helicase induced unwinding. तो हेलिकेज का काम होता है कि ओपन करना, डबल हेलिकल स्ट्रक्चर को ओपन करना. तो मान लीजिए आपके पास एक रस्सी है और वो रस्सी आपने क्या किया, एक सीलिंग के ऊपर रख दिया, मतलब टाई किया है ना, मतलब एक जो एंड होता है रस्सी का वो बांध के रखा है ऊपर और दूसरा एंड क्या है नीचे. अभी नीचे से आप रस्सी को ओपन करो, रस्सी में दो दो ये होता है ट्विस्ट किए वाला, तो रस्सी को ओपन करो, आप खींचो, तो क्या होगा, आप एक एंड से खींच रहे हो, आप एंड से एक एंड से ओपन कर रहे हो, लेकिन दूसरे एंड पे क्या हो रहा है, दूसरे एंड पे वो उसके ऊपर है ना टॉर्सनल स्ट्रेन आ रहा है, उसके ऊपर सुपर कॉइलिंग हो रहा है ना. तो एक एंड से आप ओपन कर रहे हो, दूसरे एंड से क्या हो रहा है, सुपर कॉइलिंग हो रहा है, टॉर्सनल स्ट्रेन आ रहा है. ठीक है, और जब आप छोड़ते हो तो वो टॉर्सनल स्ट्रेन कम हो जाता है. सो एग्जैक्टली सिमिलर in the DNA replication also. So what is the role of helicase? It will open the double helix at one end, but at another end there is a super coiling. Okay. Now after the replication of DNA, particular part replicate हो गया, तो लेकिन जो स्ट्रेन आया है, जो सुपर कॉइलिंग हुआ है, वो ऑलरेडी है वहां पे. So that super coiling should be relieved, and to relieve that super coiling, the role of topoisomerase is there. So these topoisomerase, they will cut the DNA and they will leave the super coiling, and they will seal that cut end, that is a sealing. Okay. Now this enzyme is very important. Now some antibiotics, जैसे आपका डीएनए गायरेज इनहिबिटर, जैसे आपका सिप्रोफ्लोक्सिन, नॉरफ्लोक्सिन, you know, ofloxacin, what they do, they inhibit this topoisomerase enzyme in the bacteria. Okay. And they prevent the relieving of supercoiling. That's why bacteria में relieve नहीं हो रहा है. After the replication, the supercoiling, after the replication, this strain or supercoiling, whatever is supercoiling is there due to replication, so that should be relieved immediately. That is relieved by topoisomerase, and this bacterial topoisomerase is inhibited by this DNA kinase inhibitor or quinolones, and they kill the bacteria. That's all. Because this is the basis of understanding the mechanism of action, right? Now next enzyme in the DNA replication is the DNA ligase. Ligase मतलब क्या, to ligase means to tie something. So DNA ligase मतलब ये join करेगा, है ना. So it is a nick sealing enzyme, nick formed between nascent chain of Okazaki fragment on the lagging strand requires segregation. Okay. So in the double stranded DNA, one strand is known as a template strand, and one strand is known as a complementary strand. Now DNA polymerization occurs, or DNA replication occurs on the both strand, लीडिंग स्टैंड एज वेल एज लैगिंग स्टैंड. तो आपको दो स्टैंड होते हैं, एक तो टेम्पलेट होता है, एक कॉम्प्लीमेंट होता है. तो टेम्पलेट को कॉम्प्लीमेंट के बजाय टेम्पलेट कॉम्प्लीमेंट के बजाय अपन इसको बोलेंगे लीडिंग स्टैंड एंड लैगिंग स्टैंड. नाउ लीडिंग स्टैंड एंड लैगिंग स्टैंड. सो डीएनए synthesis, that is synthesis of DNA, means DNA replication that occurs simultaneously on the lagging strand as well as leading strand. Okay, but on the leading strand, DNA synthesis is a linear, it is a continuous. On the lagging strand, DNA synthesis is not a continuous, it is a discontinuous, because leading strand के ऊपर क्या होता है 3 dash, leading strand के ऊपर 5 dash to 3 dash end होता है, and that's why replication is continuous in that leading strand. So replication start from 5 dash end to 3 dash end. DNA is synthesized from 5 dash to 3 dash end. Okay. Now on the lagging strand, जो lagging strand होता है दूसरा, उसमें discontinuous synthesis होता है DNA का, and because of that strand is 5 dash to 3 dash, तो opposite strand होता है. तो that's why, so part में synthesis होता है, और वो जो फ्रेगमेंट्स बनते हैं उनको बोलते हैं अपन ओकाज़की फ्रेगमेंट्स, एंड वो फ्रेगमेंट बाद में लाइगेट किए जाते हैं, बाद में ज्वाइन किए जाते हैं बाय डीएनए लाइगेट्स. नाउ वी विल सी द रेप्लिकेशन प्रोसेस इन डिटेल, बट फर्स्ट ऑफ ऑल यू शुड अंडरस्टैंड द रोल, राइट? Am I audible? Am I audible to you? Yes, am I audible to you? Now next is a single stranded binding protein. ये सिंगल स्ट्रैंडेड बाइंडिंग प्रोटीन जो होता है, इसका काम क्या है? So this single stranded binding protein also will bind to the separated strand of DNA and will prevent the re-annealing of double stranded DNA. तो जो DNA हेलिकेज का काम होता है DNA को ओपन करना, और इसका काम होता है कि जो ओपन किया वाला DNA उसको फिर से रीजॉइन नहीं होने देना, क्योंकि वो किसके लिए लग रहा है, रेप्लिकेशन के लिए लग रहा है, right? So these are the different proteins and enzymes which has a very important role or key role in the replication process. So first understand this enzyme and then we'll understand the replication process. So याद रखना है आपको किसका क्या रोल है ना, हर एक का अपना अपना रोल दिया है भगवान ने। डीएनए पॉलीमरेज को रोल दिया है कि बस आपको क्या करना है, you have to catalyze the addition of nucleotide bases. डीएनए पॉलीमरेज में 3 टाइप होता है, डीएनए पॉलीमरेज 1, 2, 3। सो 1 का रोल होता है ना, अलग रोल होता है, 2 का रोल अलग है, 3 का रोल अलग है। सो डीएनए पॉलीमरेज 3 विल ओनली ऐड द न्यूक्लियोटाइड्स। ओके, डीएनए हेलिकेज का रोल होता है टू ओपन द डबल स्ट्रैंडेड डीएनए, लाइक ए जिप ओपनर। डीएनए हेलिकेज विल ओपन अ डबल स्ट्रैंडेड डीएनए, एक साइड से ओपन करेगा, दूसरी साइड के ऊपर क्या होगा, स्ट्रैंड डेवलप होगा। अपन बाद में देखेंगे थ्रू ए डायग्राम ऑल्सो। डीएनए प्राइमेज का रोल होता है टू सिंथेसाइज प्राइमर, आरएनए प्राइमर। टोपोआइसोमरेज का रोल होता है जो डीएनए हेलिकेज की वजह से जो ओपनिंग हुआ है और एक साइड पे ओपनिंग हुआ है, एक साइड पे सुपर कॉइलिंग हुआ है, तो वो सुपर कॉइलिंग रिलीव करने का काम किसका है, टोपोआइसोमरेज का। इसमें भी टाइप होता है, टोपोआइसोमरेज 1, 2 ना एंड 3। टोपोआइसोमरेज 1, 2, 3, 4 होता है। So also topoisomerase inhibitors, they are also antibiotics, antibacterial drug, and some topoisomerase inhibitor like topoisomerase 1 inhibitor, topoisomerase 3 inhibitor। 1 and 3 को इनहिबिट करने वाले जो ड्रग है, all they are the anti cancer drug। ये दोनों को जो इनहिबिट करते हैं टोपोइसोमरेज 1 and 3 को, एंटी कैंसर ड्रग, जैसे टोपोटिकैन, टोपोसाइट्स, so they are the anti cancer drugs। देन डीएनए लाइगेज का काम होता है कि जो ओकोज़की फ्रैगमेंट और जो डिसकंटिन्यूअस सिंथेसिस हुआ है ऑन द लैगिंग स्टैंड, तो उनको सील करना, उनको ना रिक्वायर्स एनर्जी, ये इनको एनर्जी लगता है एटीपी की। नाउ सिंगल स्ट्रैंड बाइंडिंग प्रोटीन का काम क्या होता है कि जो काम डीएनए हेलिकेज ने किया है ओपनिंग का ना, ओपनिंग किया है टू स्ट्रैंड्स, दे आर ओपन बाय डीएनए हेलिकेज, दे कैन बी अगेन सील्ड। So to prevent the premature re-annealing, they can annihilate themselves। वो साथ में भी आ सकते हैं, सेपरेट किया है उनको, लेकिन वो ज्वाइन भी हो सकते हैं, तो ज्वाइन ना हो इसलिए ये बाइंड करता है। They are known as DNA stabilizing protein, उनको बोलते हैं DNA stabilizing protein। वो स्ट्रक्चर को स्टेबलाइज करते हैं, यानी ये कहीं यूनाइट नहीं होने, जब तक आपका रेप्लिकेशन कंप्लीट नहीं होता तब तक। That's why single stranded binding protein or DNA destabilizing protein, destabilize। Okay, we'll see the replication of